Welcome to the deep dive. Today, we're really zeroing in on something fundamental for cells, for keeping our genome stable DNA repair.
Absolutely. It's essential stuff.
And specifically, we're going to unpack two key ways cells handle DNA damage, particularly the kind, caused by ionizing radiation. That's obviously bread and butter in radiation oncology.
Exactly. We'll be focusing on single-strand break repair, SSBR for short, and non-homologous end joining, NHEJ, which is, well, a main pathway for fixing the trickier double-strand breaks.
Right. And understanding these is, it's critical for getting how radiation affects cells and why certain cancer therapies actually work.
Couldn't agree more. It's foundational.
Okay, so let's jump right in. A single-strand break in SSB. What exactly are we talking about here?
Okay, imagine the DNA double helix, right? Like a ladder.
Yeah.
A single-strand break is like someone just snipped one of the side rails, just one. The other side, the other rail, is still intact.
And that intact strand is important.
Oh, absolutely crucial. It serves as the template, basically, for the repair we're going to talk about.
Got it. Yeah. So what causes these little nicks, these SSBs?
Well, ionizing radiation is a big one for sure. It can smash right into the DNA backbone directly.
Direct hit.
Yep. But it also works indirectly. Radiation creates these highly reactive molecules, free radicals.
Yes, the indirect effect.
Exactly. And those radicals can also damage the DNA, leading to SSBs. But it's not just radiation. No, they can happen just from DNA sort of decaying spontaneously over time, chemically, and even, believe it or not, from just normal metabolism. Our cells make reactive oxygen species, sort of like metabolic exhaust fumes.
And those can damage DNA too.
They sure can. So it's like a constant low-level attack happening all the time inside our cells.
Wow. So our DNA is really under siege constantly, okay?
Yeah.
It's just one strand broken. Why is that such a big deal then? Why worry?
Well, it's true. It's not as immediately catastrophic as breaking both strands.
Right.
SSBs can still gum up the works. They can interfere with really important processes like DNA replication, copying the DNA or transcription, reading the genes.
So it throws a wrench in things.
It can, yeah. And maybe more worrying, if you get two SSBs happening really close to each other but on opposite strands.
Uh-oh.
Yeah, then suddenly the cell's got a full-blown double-strand break on its hands. It's like a small crack in a bridge, maybe okay on its own, but if another one forms right nearby,
then you get serious structural failure potential.
Exactly. The whole thing could break.
That makes perfect sense. So how does the cell fix these single-strand breaks? Let's walk through SSBR.
Okay, step one is crucial. Sensing the damage and kicking off a signal.
The alarm system.
Pretty much. And the main player here, the first responder, is a protein called PRP1. It's incredibly fast and efficient.
PRP1.
PRP1 basically zips over and binds right onto that single strand break. As soon as it binds, it switches on. And what it does then is pretty cool. It starts adding these long chains of a molecule called poly-ADP ribose, or PAR. PAR, yeah. It adds these PR chains to itself and to other proteins nearby. Think of it like PRP1 isn't just seeing the break, it's like energetically waving this huge molecular flag.
A beacon.
A beacon, exactly. A big, bright flag made of PR shouting, "Damage here, repair crew needed." This parylation, as it's called, attracts all the other necessary repair factors right to the spot.
Wow. That's quite a signal, and you said it's fast.
Super fast, which is good given how often these SSBs happen.
Okay, so PRP1 raises the alarm, waves the flag. What's next?
Next, you need to bring in the scaffolding, get the site organized.
Right.
And this is where a protein called XRCC1 comes in. We actually see this protein in another repair pathway, too. Base excision repair, or BER. It's kind of a versatile manager for single-strand damage.
XRCC1. Like a molecular toolkit or maybe a foreman?
Foreman's a great analogy. It recognizes that PR signal from PRP1, binds there, and then acts as this platform, this coordinator, to bring in all the specific enzymes needed for the next steps.
Makes sense. Get the right tools to the job site.
Precisely. Now, often, especially with radiation damage, the broken ends of the DNA they're not clean.
Dirty ends.
Yeah, you could call them that. They might have damaged sugar bits or phosphate groups left behind. They're not ready to be just stitched back together.
So cleanup crew needed.
Exactly. And that's often the job of an enzyme called polynucleotide kinase phosphatase, or PNK.
PNK.
PNK comes in and tidies up those messy 3' and 5' ends. It processes them so they're chemically ready for the next steps, synthesis and ligation.
Okay.
And, sometimes near the break, there might be a spot where the base itself is just missing an abasic site. If that's the case, another enzyme we know from BER called APE1 might get involved to snip the backbone there too.
So it ties into other repair systems sometimes.
Definitely overlap. Very efficient use of resources by the cell.
Okay, so we've spotted the damage with PRP1, brought in the formin XRCC1, cleaned up the ends with PNK. Now we actually had to fill the gap, right?
Exactly. Patch the hole. And there are basically two main ways this happens in SSBR.
Two subpathways.
Kind of. There's short patch SSBR and long patch SSBR.
Okay, short patch first.
Short patch is usually for simpler breaks, maybe just one missing or damaged nucleotide. In that case, DNA polymerase beta, another player from BER, comes in, adds the single correct nucleotide.
Fills the tiny gap.
Yep. And then DNA ligase III seals the final nick in the backbone. And XRCC1 is often right there coordinating this final step. Very neat.
Okay, that sounds pretty straightforward. What about long patch?
Long patch SSBR gets used for more complex damage, where maybe you need to replace several nucleotides.
Bigger patch needed.
Right. So here, different polymerases get involved, usually DNA polymerase delta or epsilon. They work with other helper proteins, PCNA and RFC.
The replication machinery components.
Some of them, yeah. They synthesize a longer stretch of new DNA, maybe 2 to 10 nucleotides or so. And as they synthesize, they actually push the old damaged strand segment out of the way, creating a little flap.
A flap of old DNA.
Exactly. Then another enzyme, flap endonuclease 1, or FEN1, comes along and cuts off that flap.
Snips the excess.
Precisely. And then finally, a different ligase, DNA ligase I, seals the last remaining nick. So it's a bit more involved, longer synthesis, flap removal, than sealing.
Wow, that is remarkably precise. Amazing molecular machines at work. And you mentioned the overlap with base excision repair quite a bit.
Yeah, it's significant. Sharing enzymes like XRCC1, pol beta, PNK, APE1, the ligases. It really shows how the cell handles different kinds of single-strand damage efficiently.
Makes sense. Reuse the tools you have.
Exactly. And given how incredibly common SSBs are, this robust, somewhat overlapping system is just absolutely vital for keeping our genetic code intact, day in, day out.
Okay, that covers the frequent but usually manageable single-strand breaks. Now, let's shift gears to what you call the big problem, double-strand breaks, DSBs.
Right, the big one.
Why are these so much more critical, especially for us thinking about radiation effects?
Well, back to the latter analogy, a DSB isn't just one rail snipped, it's both rails completely severed, broken right through.
The whole ladder is in two pieces.
Essentially, yes. Now, think about frequency. After a typical radiation dose, say 1 gray, a cell might get around maybe 1,000 SSBs.
1,000.
Yeah, but only about 40 DSBs.
40. Okay, much less frequent, but you're saying they're way more dangerous.
Way more dangerous, for a few reasons. First, a DSB can literally cause a chromosome to break into separate pieces. You could lose huge chunks of genetic information or end up with unstable bits floating around.
That sounds bad.
It is. Second, unlike an SSB where you have that intact opposite strand as a perfect blueprint for repair.
Right, the template.
With the DSB, that immediate template is gone. Both strands are broken at the same spot, so accurate repair becomes a much bigger challenge.
Yep, makes sense. No easy instructions.
Exactly. And maybe most importantly for radiation oncology, if these DSBs aren't repaired or if they're repaired incorrectly, that's a major way radiation actually kills cells. It prevents them from dividing successfully.
Cell kill, the goal of therapy often.
Right. But misrepair can also lead to nasty chromosomal problems, deletions, translocations where bits of chromosomes get stuffed to the wrong ones, dicentrics, ring chromosomes.
Those sound like recipes for genomic chaos.
They really are. These aberrations can destabilize the entire genome and potentially, down the line, even contribute to cancer development itself if the cell survives. So DSBs are serious business.
Definitely the big problem. Okay, so how does the cell even begin to tackle something like this?
Well, it's got two main strategies, two major pathways for DSB repair. The first one, our focus today, is non-homologous end joining, NHEJ.
NHEJ.
Think of NHEJ as the cell's emergency response team. It's fast, but maybe a bit rough and ready.
Quick and dirty.
That's the classic description, yeah. It basically grabs the two broken ends and, well, sticks them back together. Speed is often the priority. But because it's fast and doesn't use a detailed template, it's more likely to make small errors right at the break site.
Okay, so speed over absolute perfection sometimes. What's the other pathway?
The other main one is homologous recombination, or HR. This one is much slower, much more complex, but it's incredibly accurate.
High fidelity.
Exactly,
Yeah.
because it uses the undamaged sister chromatid, the identical copy of the chromosome that's present after DNA replication, as a perfect template to guide the repair.
Ah, okay. So it needs that copy.
It does, which means HR is mostly active in the S and G2 phases of the cell cycle after the DNA has been copied.
Got it. But we'll dive deeper into HR another time.
Definitely. It deserves its own deep dive. Today, it's all about NHEJ.
Okay, NHEJ. The name itself, non-homologous end joining, tells us something.
It really does. Non-homologous means it doesn't need that identical template that's sister chromatid like HR does. It can work even if the ends aren't perfectly matched or if there's no template nearby.
Right.
And end joining is pretty literal. It directly joins or ligates the broken ends back together.
But you said it's error-prone. How does that happen?
Well, because it doesn't rely on a precise template and because the broken ends often need some tidying up before they can be joined.
The dirty ends again.
Yeah. That tidying process can sometimes involve trimming back a few DNA bases from the ends, or sometimes small random bases might get inserted. So it's very common to end up with small insertions or deletions, people call them indels, right at the spot where the break used to be.
Ah, so the repair leaves a tiny scar, potentially altering the sequence slightly.
Exactly. That's the trade-off. You get speed, which can be vital to just hold the chromosome together, but you might sacrifice a little bit of sequence accuracy right at the junction.
But despite this potential for mistakes, NHEJ is the main pathway in many cases.
That's absolutely right. In mammalian cells like ours, NHEJ is the dominant DSB repair pathway for much of the cell cycle, particularly in G1 before the DNA gets replicated, so there's no sister chromatid available for HR anyway.
So most of the time, if a cell gets a DSB, it's NHEJ that jumps in first.
Generally, yes. And it's not just for radiation damage. NHEJ is also absolutely essential for a normal process in our immune system called VDJ recombination.
What's that?
It's how our developing B cells and T cells actually cut and paste gene segments together to create the massive diversity of antibodies and T cell receptors we need to fight off infections. That cutting process deliberately creates DSBs.
And NHEJ stitches them back together.
Exactly. It stitches them back together in new combinations. So this quick and dirty repair pathway is fundamental not only for fixing accidental damage, but also for building our entire adaptive immune defense. It's quite remarkable.
Wow, that's fascinating. A repair mechanism moonlighting in immune system construction. Okay, let's get into the nuts and bolts of NHEJ then. How does it start sensing the break?
Right, damage sensing. Now, other proteins like the mRN complex and the ATM kinase definitely get involved early, signaling the presence of a DSB, but the real workhorse for grabbing onto the broken ends in NHEJ is the Ku heterodimer.
Ku. Okay, tell us about Ku.
Ku is a protein complex made of two parts, Ku70 and Ku80. You can picture it like a ring or maybe a molecular clamp.
The clamp, I like that.
It has this amazing ability to recognize and bind very quickly to the raw ends of a DNA double-strand break. It slides onto each broken end,
Grabs both pieces.
grabs both pieces, and essentially holds them close together. This does a couple of things. It protects the ends from getting chewed back too much by other enzymes.
Stabilizes them.
Yes,
Right.
and crucially, Ku acts as a landing pad, a scaffold, to recruit the rest of the NHEJ machinery to the site.
So Ku is the first responder and the foreman for NHEJ.
In a way, yes. It gets there first, holds things together, and calls in the rest of the team.
Okay, who does Ku call in next?
Its main partner is a very large protein kinase called DNA PKCs. That stands for DNA-dependent protein kinase catalytic subunit.
DNA PKCs, quite a mouthful.
It is, but it's central. Ku bound to the DNA ends recruits DNA PKCs, and when DNA PKCs binds to the Ku DNA complex, its kinase activity gets switched on.
Kinase. So it adds phosphate groups to things.
Exactly. It phosphorylates itself, it phosphorylates Ku, and it phosphorylates other NHEJ proteins that arrive later. This wave of phosphorylation is like flipping a series of switches, coordinating the whole repair process.
Okay, so Ku grabs the ends, recruits DNA PKCs, which then activates everything by phosphorylation. What happens after activation?
Now we get to that end processing part we mentioned, dealing with those dirty or incompatible ends.
Right, the cleanup.
The key enzyme for this in NHEJ is called Artemis.
Artemis, like the Greek goddess.
Indeed. And Artemis is a nucleus. It gets recruited to the break site by the DNA PK complex, and its activity is actually turned on by DNA PKC's phosphorylation.
So DNA PK activates the cleaner upper.
That's right. Artemis has abilities to cut within a DNA strand, endonucleus, and chew back from the ends, exonucleus. This lets it trim off any awkward overhangs, remove damaged bases, basically make the ends ligatable, ready to be joined.
But this is where the errors can creep in.
This is a major source of them, yes, because that trimming action can remove nucleotides from the original sequence. So you often end up with deletions right where Artemis did its work.
Necessary but potentially messy. Does anything else help clean the ends?
PNK, the enzyme we met in SSBR, might also play a role here sometimes, helping to prepare the ends chemically for ligation.
Okay, so Artemis and maybe PNK process the ends. What then? Is there any gap filling like in SSBR?
Sometimes, yes. If after Artemis's work there are still small gaps, maybe just one or two nucleotides, specialized DNA polymerases can fill them in. These are polymerase mu and polymerase lambda.
Different polymerases than in SSBR.
Yes, these belong to the PolX family. They're a bit unusual because they can sometimes add bases even without a template, or they might misread a template. So they can also contribute to the small insertions or sequence changes sometimes seen at NHEJ junctions.
Adding a bit more potential variation.
Potentially, yes.
Right. Ends are processed, small gaps might be filled. Now for the final step, stitching it all up.
Exactly. The final ligation. Sealing the breaks in the DNA backbone. This is the job of the DNA ligase 5E complex.
Ligase 5, is it working alone?
No, it has essential partners. It forms a stable complex with another protein called XRCC4, which acts as a scaffold.
XRCC4.
Yeah.
Sounds familiar.
Different from XRCC1 and SSBR, but same naming idea. And there's also an accessory factor called XLF, or sometimes Cernunnos.
XLF.
So it's this whole complex, ligase 4, XRCC4, and XLF, that performs the final chemical reaction, creating the phosphodiester bonds to rejoin the DNA strands and complete the repair.
A multi-protein machine for the final seal.
That's right.
Okay, that seems like a pretty complete pathway. Ku, DNA PKCs, Artemis, maybe Pol Lambda, and the leak safety complex. Does that, everyone? You mentioned 53BP1 earlier, too.
Ah, yes, 53BP1. Good catch. It's not part of the core joining machinery itself, but it's a really important regulator.
How does it regulate?
53BP1 plays a big role in promoting NHEJ, especially in the G1 phase. One key thing it does is it binds near the break site and helps to prevent the DNA ends from being excessively chewed back or resected.
Why prevent resection?
Because extensive resection is the necessary first step for the other repair pathway, homologous recombination.
Ah, so 53BP1 puts the brakes on HR.
It helps to, yes. By limiting resection, especially in G1 when there's no sister chromatid for HR anyway, 53BP1 effectively channels the repair towards NHEJ. It's like a traffic controller, directing DSBs down the NHEJ route when appropriate.
Fascinating. Influencing the pathway choice. So that core NHEJ team, Ku grabbing ends, DNA PKCs activating, Artemis processing, ligase VA complex sealing. Any easy way to remember them?
There's a mnemonic some people use. Kids always dream late XL.
Kids always dream late XL. Let's see. K for Ku.
K for Artemis.
D for DNA PKCs.
Mark for ligase IV.
And XL for XLF. That's actually pretty handy.
Can help keep the main players straight.
Great. Now, this is all fascinating molecular biology, but let's bring it right back to the clinic, the clinical relevance. So important for us in Rad on TEC. What happens when these pathways go wrong? Let's start with SSBR defects.
Okay. SSBR and PRP inhibitors. This is a huge success story in targeted therapy. Remember, PRP1 is that key sensor in SSBR.
Right, waving the flag.
Okay, now think about certain cancers, especially some breast and ovarian cancers, that have mutations in genes like BRCA1 or BRCA2.
The BRCA genes, they're involved in homologous recombination, right?
Exactly. They're crucial for the high-fidelity HR pathway. So, tumor cells with BRCA mutations have defective HR. They can't repair DSBs accurately using that pathway.
So they're crippled in one major repair system.
Precisely. And that makes them become highly dependent on other pathways to survive, including SSBR, which relies on PRP.
Ah, they lean more heavily on PRP and SSBR because their HR is broken.
You got it. This sets up a vulnerability. It's called synthetic lethality. The tumor cell can tolerate losing HR, or it could tolerate losing PRP-mediated SSBR, but it can't tolerate losing both.
So if you knock out the remaining pathway.
If you treat these BRCA mutant HR-deficient tumor cells with a drug that inhibits PRP.
A PRP inhibitor.
You block their main remaining way of dealing with all those common single-strand breaks. Those unrepaired SSBs then often turn into DSBs when the cell tries to replicate its DNA. And now the cell has a DSB problem it can't fix properly because its HR is already broken.
And the cell dies.
The tumor cell dies. That's the basis for using PRP inhibitors so effectively in BRCA-mutated cancers. We're exploiting their specific DNA repair defect. It's a beautiful example of translating basic science understanding into powerful therapy.
Really elegant. Leveraging the cell's own weaknesses.
Yeah.
Okay, what about defects in the NHEJ pathway itself? What are the clinical consequences there?
Right. Since NHEJ is needed for both fixing radiation damage, DSBs, and for that VDJ process in the immune system.
The antibody and T-cell receptor building.
Yes. Inherited mutations in the genes for key NHEJ proteins like Ligascorly, XRCC4, XLF, DNA PKCs, or Artemis cause some really severe human syndromes.
All kind of syndromes.
They typically present with severe combined immunodeficiency, SCID.
SCID, like the bubble boy disease.
Yeah, that kind of severity, yes. Because without functional NHEJ, the VDJ recombination fails, and the person can't generate the diverse immune cells needed to fight infections.
Profound immunodeficiency.
Critically for us, these patients also show extreme radiosensitivity.
Makes sense. They can't fix the DSBs from radiation.
Exactly. Their cells are incredibly sensitive to killing by ionizing radiation. So patients with conditions like LIG4 syndrome or Artemis-deficient SCID, sometimes called Athabascan SCID, giving them standard doses of radiation therapy could be catastrophic, causing devastating toxicity.
So recognizing these rare syndromes is crucial before starting radiation.
Absolutely vital. It highlights how fundamental NHEJ is for both normal immune development and for handling the type of DNA damage we rely on in radiation therapy.
Really drives home the importance of these pathways. Okay, let's crystallize some key takeaways. Our clinical pearls for listeners, especially residents.
Sounds good. Pearl number 1. Remember, PRP1 initiates SSBR, and in tumors with broken HR, like BRCA mutants, they become addicted to PRP. That's why PRP inhibitors work synthetic lethality.
Got it. PRP inhibitors exploit HR deficiency via SSBR reliance. My turn for a pearl. NHEJ is the main DSB repair pathway, especially in G1. It's fast, but remember it's error-prone, think Adele's. This quick and dirty nature has implications for genomic stability long-term.
Excellent point. And final pearl for me. Inherited defects in NHEJ core proteins, think Ku, Artemis, DNA PKCs, ligase-Cy-B complex cause radiosensitive SCID. Be incredibly cautious with radiation in patients with unexplained SCID or known NHEJ defect.
Crucial safety point. Okay, ready to test ourselves? Time for our board review section.
Let's do it. Question 1. Which protein complex acts as the primary sensor and scaffold at DNA double-strand break ends to initiate non-homologous end joining NHEJ? Is it A, mRN complex, B, polymerase mu or lambda, C, Q70 and Q80 heterodimer, or D, Ligas 4 XRCC4 XLF complex?
Primary sensor and scaffold for NHEJ. That's got to be Q. It binds the ends first and brings everything else in. So CQ70 and Q80 heterodimer.
Spot on. Rationale. Q binds rapidly to the broken ends and recruits the downstream factors. Perfect. Okay, question 2. A patient is diagnosed with a rare genetic disorder. They have severe combined immunodeficiency, SCID, and their cells are extremely sensitive to ionizing radiation, a mutation in a gene for a key component of which DNA repair pathway is the most likely cause. A, base excision repair, BER, B, nucleotide excision repair, NER, C, non-homologous end joining, NHEJ, or D, mismatch repair,
MMR.
Okay, SCID plus radiosensitivity, that combo screams NHEJ defect because NHEJ is needed for both VDJ recombination immune system and DSB repair radiation. So definitely C, non-homologous end joining, NHEJ. Correct again. Rationale. NHEJ's dual role in DSB repair and VDJ recombination links radiosensitivity and SCID. All right, question 3. Which enzyme is primarily responsible for processing potentially incompatible or dirty DNA ends during NHEJ, but also contributes to the error-prone nature of the pathway? A, polymerase beta, B, APE1, C, Artemis, or D, FEN1?
Processing dirty ends in NHEJ and potentially causing errors. That sounds like Artemis the nucleus. So C, Artemis.
Excellent. Rationale. Artemis is the nucleus that cleans up the ends for ligation in NHEJ, but its trimming activity is a major source of indels. Last one, question 4. PARP1 plays a critical early role in which DNA repair pathway? A, nucleotide excision repair, NER, B, non-homologous end joining, NHEJ, C, homologous recombination HR, or D, single-strand break repair, SSBR.
Perfect. Rationale. PROP1 is the key initial sensor for single-strand breaks, kicking off the repair cascade. Great job.
Okay, good review. So let's quickly recap the big picture from today. Yeah, so we covered single-strand break repair, dealing with those common nicks, key players, PRP1 sensing it, XRCC1 coordinating, enzymes like PNK cleaning ends, pol beta or pol delta epsilon filling gaps, and ligases sealing it up. And remember, the PRP inhibitor connection to HR-deficient tumors.
Right, and then the big one, double-strand breaks. We focused on NHEJ, the fast, dominant, but error-prone pathway, especially in G1.
Key NHEJ team, coup grabbing ends, DNA PKCs activating, Artemis processing, the messy part, and the Ligas IV XRCC4 XLF complex sealing the deal, and 53 BP1 helping to steer traffic towards NHEJ.
And crucially, defects in NHEJ lead to that dangerous combination of radiosensitivity and SCID.
Exactly. It connects the basic science directly to patient safety and treatment strategies in radiation oncology.
Absolutely. So that covers SSBR and the quick and dirty NHEJ pathway. Next time on the deep dive, we'll tackle the other side of the DSB coin, the high-fidelity pathway, homologous recombination, HR.
Yeah, understanding how NHEJ and HR work together, how the cell chooses between them, that's really key to getting the full picture of the DNA damage response.
It really is. Understanding these fundamental processes, well, it underpins so much of what we do, doesn't it?
It really does. It informs how we think about therapy, toxicity, everything.
Well, we hope this deep dive into SSBR and NHEJ has been useful and maybe even a little clearer now. Thanks so much for joining us.
Yeah, thanks everyone. Keep exploring this stuff. It's endlessly fascinating and important.